Hello friends, Ace Botany next episode. So in this episode, we'll be discussing cell, the unit of life, fourth one, chapter number 8. So what all things we'll be discussing in this episode? For all that, I will share the screen so you can find the note also. I think screen came, so we can proceed to discuss cell, the basic unit of life, fourth episode, chapter number 8. And what all things we'll be discussing as usual, there will be an introduction. Then endomembrane system, including endoplasmic reticulum, Golgi body, Golgi apparatus, GB or GA, lysosome and vacuole. After that, 5 theory question, 5 NEET model question with the key as usual. So we can proceed to introduction. So in this episode, we will start to discuss the cell organelles of eukaryotic cell, starting with the endomembrane system, then questions as usual. So what is meant by endomembrane system? Theory question, NEET model question. So when we try to explain endomembrane system, in eukaryotic cell, some membrane-bound cell organelles are considered together as endomembrane system. So automatically, why? Why this cell organ selected? Because their functions are coordinated. And these cell organs include endoplasmic reticulum, Golgi apparatus or Golgi body, lysosome and vacuole. I think first page is clear. Now we can proceed to second page with the endoplasmic reticulum. This is the first cell organ. In this section we have to learn endoplasmic reticulum, eukaryotic cell, where we can find first two diagrams. These two are old NCERT. This is present NCERT diagram. Present one, labeling question, identifying question, etc. Now we can further proceed to endoplasmic reticulum. So what is the meaning of the term endoplasmic reticulum? Endoplasm always means inner. That is relatively fluid part of the cytoplasm. Reticulum means a network-like structure, reticulum. In algae, you may be studied reticulate chloroplast. So network. Now here, main scientist involved. Discovery is concerned with the scientist Keith Porter, 1945. Similarly, one more is there, Helen Thompson, 1945. Name given by Keith Porter, 1953. Another name was given by Garner, namely ergastoplasm. In animal cell, muscle cell, it is called sarcoplasmic reticulum. Now, other side of the diagram, everything given this will be easy for you. It is connecting, that is endoplasmic reticulum is connecting plasma membrane to nuclear membrane at certain places. It is a interconnected network-like system. Diagram given NCERT. Now, basically, what are the components of endoplasmic reticulum or ER? 3 types of structures we have to learn: cisternae, vesicles, and tubules. So we can proceed to it. So 3 diagrams I drawn there, cisternae, vesicles, and tubules. You can find them. First item, cisternae. Cisternae are flattened, long, parallel, interconnected, sac-like structure. Cisternae. This is a diagram you can find here. Now, mainly seen in cells which are involved in synthetic activities like protein synthesis, as in cells of brain and pancreas. Normally associated with the ribosomes, larger subunit 60S. Eukaryotic ribosome. Now comes second component is tubules. So cisternae, tubules, and vesicles. So second one is tubules. Tubules are tube-like nature, branched or unbranched, forming a reticular system by connecting with the cisternae and vesicles. ER tubules generally lack ribosome, and they are usually seen in cells involved in steroid or lipid synthesis, tubules. Now comes third term, vesicles. Vesicles are round or oval in shape. Ribosomes generally absent, abundant in cells like pancreas, also seen in spermatocytes. Endoplasmic reticulum vesicles related to microsome. There is a structure called microsome. It is connected with endoplasmic reticulum vesicles. I think up to that it is clear. Now division made by endoplasmic reticulum. See, in the intracellular space, ER makes 2 parts. That is, since components of ER is scattered in the cytoplasm, ER divides the intracellular within space into 2 distinct compartments. One is luminal. Luminal means inside endoplasmic reticulum. Other is extraluminal, that is rest of the cytoplasm. Luminal means basically internal space enclosed by ER membrane. While extraluminal automatically basically means rest of the cytoplasm excluding ER part. Luminal and extraluminal. Sorry. 2 types of ER based on ribosome. Another important item. One is smooth endoplasmic reticulum, otherwise called SCR. Second is rough endoplasmic reticulum, otherwise called ARIA. NCERT figure, photograph given there, labeling question, SCR, ARIA, all this. Now, smooth endoplasmic reticulum, otherwise called SCR. SCR is same as egg granular because ribosomes will not be there. So, egg granular endoplasmic reticulum since no ribosomes attached. Sarcoplasmic reticulum is the smooth endoplasmic reticulum in muscle cells. Now comes SCR main functions. So many functions are there. Basically, 4 5 we have to discuss. Linked with preferably NCERT. It is a major site for lipid synthesis, sterols, hormones, formation of spirosome, muscle stores Ca2 plus ions for release during muscle contraction. Then detoxification of toxic chemicals with the help of cytochrome P450. Synthetic products of ARIA passes to Golgi complex through SCR. Synthetic products of ARIA passes to Golgi complex through SCR. Some main functions given. Still many are there, but that is far away from NCERT. Now comes rough endoplasmic reticulum, otherwise called ARIA. ARIA is same as granular endoplasmic reticulum because ribosomes will appear like granules, since ribosomes are seen on surface. For attaching with the ribosome, ARIA contains two types of glycoprotein, important for entrance point of view. One is riboforin 1 and other is riboforin 2, connected to 60S subunit of 80S ribosome, because we are dealing with the eukaryotes, connected to 60S subunits of 80S ribosome. ARIA is rich in cells active in protein synthesis, secretory cells. In association with the Golgi apparatus, ARIA helps to produce lysosome. SCR can develop from ARIA. So all are linked endomembrane system. Now comes second organelle for us. It is Golgi apparatus. First two diagram, previous NCERT diagram, just for reference, I shown that. Third is present NCERT diagram. Fourth is just represented by drawing and parts are labeled there. Now we can proceed to Golgi apparatus, Golgi complex, etc. To connect with the Golgi apparatus, Golgi body even, scientists involved, first seen by George A. in 1867, named after Italian scientist Camillo Golgi, 1898. Other names for Golgi apparatus, Golgi complex, Dalton complex, apparato reticule, etc. According to Golgi, Camillo Golgi, it is a reticular structure located near to the nucleus. Initial study that is connected to the nerve cells of barn, owl, and cat, and method utilized were metallic impregnation. Now occurrence, where we can find it, where it will be absent. It is a eukaryotic structure, so automatically absent in prokaryote. In eukaryote itself, we can find it is absent in mature sieve tubes of plants, bryophyte and pteridophyte sperm cells, mature RBC of mammals. Dictyosome is the term often used in plants, and Golgi apparatus is made up of unconnected units. Now structure. When we come to Golgi apparatus, 4 components will be there: cisterna, vesicles, tubules, and Golgi and vacuole. We can proceed to next page. So diagram drawn. So you can easily find the label structure is all this. Left diagram is NCERT diagram. Now comes cisternae. Cisternae are flattened structure, sac-like in appearance, stacked one on another, one above other. 4 to 6 layers can be seen. Normally 4 to 6 in number. Unicisternal dictyosomes are seen in fungus. Unicisternal. Usually cisternae arranged in a concentric manner. Diagram you can find them having polarity. Convex phase, which is otherwise called cis phase, otherwise called forming phase, and concave phase, otherwise called trans phase or maturing phase. And cis and trans phases are entirely different, but the phases are interconnected. And action is cis phase will receive the content like transitional vesicle from ER. After different procedures reaches the maturing phase and budded off. I think how this is phase works. Now comes vesicles or vesicles. Vesicles are seen at the edges of the cisternae. If you check the diagram, you can find. Yeah. Edges of the cisternae, they pinch off from tubules. The vesicles are 2 types: smooth surface, smooth vesicle, rough surface, coated vesicles. Next comes tubules. So when we proceed with the tubules are interconnecting structures of different cisternae, and they are small and flat. Theoretical aspect, all this. Now, fourth is Golgian vacuoles. Golgian vacuoles are expanded spherical vacuoles developed at maturing or concave phase. It's important. Maturing or concave phase. Then they contain certain amorphous or granular-like substances. Some of Golgian vacuoles functions as lysosome important. Some of the Golgian vacuoles functions as lysosome. Now, what are the functions of Golgi apparatus? See, like other cell structure, Golgi apparatus do various functions. Some of them we will be discussing connected to the NCERT, including first is a term familiar to us, PPT. Means here, process, package, transport connected with the secretions. The usage is within or to extracellular targets. Pathway is from endoplasmic reticulum to Golgi body as transitional vesicles, fuse with the cis phase, move to trans phase of Golgi apparatus. Thus, we can find ER and GA. ER means endoplasmic reticulum, GA means Golgi apparatus, closely associated in structural and functional aspect, theory and runs question. Second function, proteins produced by ribosomes on ER to GA before they are released from its trans phase. So GA proceeds. That is concerned with the protein produced by ribosome on ER. Now, third item important for exam, glycosylation and glycosidation, important for many crucial reactions of body. So glycosylation, glycoproteins, that is glycosylation of proteins, sugar to proteins. Glycosidation, glycolipids, glycosidation of lipids concerned with the sugar to lipids. Then formation of acrosome on sperms, here you can find acrosome. So you may be studied. Membrane formation during cytokinesis, lubrication of root tip while piercing. That is regarding Golgi body. So endoplasmic reticulum, Golgi body. Now comes third one, lysosome. Main scientist is Christian de Duve. The pronunciation is different. Belgian scientist, Nobel Prize in 1964. Synonym for lysosome, suicidal bag, polymorphic cell organelle, etc. It is formed from Golgi body. Up to that, I think it is clear. This will be a rough diagram of lysosome. This is sperm head, acrosome, etc. Golgi body. Now we can proceed to next item. So usually, generally, it is lysosome is single membrane. They are rich in hydrolytic enzyme, important acid hydrolysis like proteases, lipases, carbohydrases. And for this, acidic condition is a must for their work. Optimally active at the acidic pH. So automatically question how acidity will be maintained. That is by pumping H+ ions into it. Now, a small literature you can find that was connected with the Nobel Prize. In 2016, Nobel Prize was given to Yoshinori Oshumi and et al. In connection with the discoveries of the mechanism for autophagy connected with the lysosome. Now, four forms of lysosome, that is why it is called polymorphic cell organ, primary, secondary, residual bodies, autophagic vacuoles. See, scientists believe that lysosome to be formed by the joint activity of endoplasmic reticulum, Golgi body, then lysosome. So there is a system called GERL, Golgi body endoplasmic reticulum lysosome system. Now, primary lysosome are newly formed structure at trans phase. So it will be formed in trans phase of Golgi body. It contains inactive enzyme. Secondary lysosome, which is otherwise called heterophagosome, digestive vacuole. It is formed when fusion between food-containing phagosomes with the lysosome takes place. Hydrolytic enzyme to digest it. Third structure is residual bodies, which is otherwise called tertiary lysosome. Go secondary over, now tertiary lysosome. These are lysosome in which only indigestible food materials have left. Thus, residual bodies pass out and will fuse with the cell membrane to throw the content to outwards, effaggy or exocytosis. Now, fourth is autophagic vacuoles, that is autophagosome or autolysosome. They are formed by the assembly of many primary lysosome around old or worn out or degenerate intracellular organelles. They are surrounded by vacuolar membrane and digested by autodigestion or autolysis, commonly called suicidal bags. Now, disappearance of larval structure during metamorphosis. Typical example is tail in frog due to autolysis. Now, functions of lysosome, it is extracellular digestion, autophagy. Now, you may be studying zoology at term sperm lysin lysosomal enzyme helps to break out the limiting membranes of egg. Now, some diseases connected with the lysosome. See, lysosome is not functioning in a proper manner. It can be connected with or can cause many diseases, includes Farber's disease, Hunter's syndrome, Niemann-Pick disease, polynephritis, gout, lung fibrosis, arthritic joints. Even it can lead to, it in the cells, lysosomal activity can lead to connected with the malfunctioning, aging of cells, etc. Now comes the last item for us, it is vacuoles. So what is vacuoles? Vacuoles is a membrane-bound space found in cytoplasm. Vacuoles are, within inverted comma, we have to say, considered as a non-cytoplasmic area within a cell. It possesses sap, water, excretory products, other materials not useful for the cell. Now, next is the important question. Tonoplast is the vacuolar membrane. It is semi-permeable, do transport even against concentration gradient into the vacuole, showing concentration in the vacuole is higher than the cytoplasm. So, tonoplast, vacuolar membrane, then that is semi-permeable, do transport even against concentration gradient into the vacuole, showing concentration in the vacuole is higher than cytoplasm. It's important one. Next is vacuolar formation. Scientists suggest vacuoles are formed by fusion of smaller membrane-bound vesicles, can originate from endoplasmic reticulum, Golgi body, even plasma membrane, even by engulfing food, that is forming food vacuoles. And different types of vacuoles, we have to proceed with the three. One is contractile vacuoles. In amoeba, connected with the osmoregulation, respiration, important one, exam point of view. Next is food vacuoles. In protist, food vacuole is formed by engulfing the food particles. And third is gas vacuoles, otherwise called pseudo vacuoles. Why it is pseudo, I will explain. It is seen in prokaryote. True membrane not seen. True membrane not seen. Connected with the buoyancy. True membrane not seen. Covering may be there. Connected with the buoyancy. After that, 5 theory question, 5 NEET model question with the key as usual. So what all things we learned in this episode? See, this area, cell the basic unit of life. Now already you know it is a theoretical chapter. Important for theory and entrance. Questions will be there. So introduction, endomembrane system, 4 cell organs and question. Now first we define what is endomembrane system, why the name, what is the role of functions. Then endoplasmic reticulum, 4 different photographs, of which 3 different photographs, one belongs to present NCERT, other 2 old NCERT. Then endoplasmic reticulum expanded with the meaning of the term endoplasm reticulum. Then main scientist Porter. Then ergastoplasm by Garner. Then connecting plasma membrane to nuclear membrane, so pathway, etc. Then 3 components of endoplasmic reticulum, that is cisternae, tubules, and vesicles. Cisternae, what are the characters? Then tubules, what are the characters? Vesicles, what are the characters? Then division of intracellular space by ear. That is, since components of ear is scattered in the cytoplasm, ear divides intracellular space into 2 distinct compartments, luminal inside ear, extraluminal rest of the cytoplasm. Then 2 types of ear based on ribosome, smooth SER, then rough RER. Smooth is otherwise called agranular, since ribosome is absent. Then sarcoplasmic reticulum is a smooth ER in muscle cell. Then SER main function, lipid synthesis, spirosome formation, muscle stores CA2 plus released during muscle contraction, detoxification. Then synthetic products of RER passes to Golgi complex through SER. Then rough endoplasmic reticulum RER, why it is rough, what is other name granular. Then how ribosome get attached to endoplasmic reticulum, riboform 1 and 2 connected to 60S subunit of 80SR ribosome. Then RER where we can find, then what is the function, etc. Protein synthesis mainly, secretory cells. Then SER can develop from RER, etc. RER helps to produce a lysosome also in association with the Golgi apparatus. Then Golgi apparatus again, some old NCERT diagram, then a new NCERT diagram, then a drawn diagram. Golgi apparatus, that is main scientist Camillo Golgi. Then many other names for this one, like Golgi complex, Golgi apparatus, Dalton complex apparatus, reticular, etc. Then which method? Metallic impregnation. Then located near the nucleus, the nerve cells of one or and cat occurrence where we can find in eukaryote. Prokaryote is absent in eukaryote itself, where it is absent. What is done by dictyosome and 4 structures connected with the Golgi apparatus, cisternae, vesicles, tubules, Golgian vacuoles. Then what are the features of cisternae? Cis and trans phase. Then vesicles, what is the structure? Then tubules, again structure. Then fourth one, it is Golgi vacuoles, that is can function as a lysosome, then functions of Golgi apparatus, PPT. Then pathways from endoplasmic reticulum, Golgi apparatus, transcendental vesicle fused with the cis phase and move to trans phase of Golgi apparatus. ER and GA very closely packed in structural and functional aspect. Proteins from ER to GA before release from trans phase. Then glycosylation, glycosidation, acrosome of the sperms, membrane formation during cytokinesis, lubrication of protein. Then lysosome, Christine Dedouet, Belgian scientist, Nobel Prize 1964. Then synonym, suicidal bags, polymorphic cell organ, found from Golgi body, single membrane, rich in hydrolytic enzyme, acid hydrolysis, proteases, lipases, carbohydrates, acidic condition, maintained by pumping H+ ions. 2016 Nobel Prize was given in this context. Lysosome connected with autophagy, 4 forms of lysosome, primary, secondary, residual bodies, autophagic vacuole, polymorphic. Then scientists believe lysosome to be formed by the joint activity of endoplasmic reticulum Golgi body, and then lysosome forms. So GERL system, primary lysosome, what it is, secondary lysosome, how it forms, then residual bodies, and finally autophagic vacuoles. Finally, autophagic vacuoles. Otherwise, autophagosome or autolysosome. Then disappearance of larval structure during metamorphosis tail in frog due to autolysis. Functions of lysosome. This is connected with the lysosome. Then vacuoles. What is meant by vacuole? How it is formed? Vacuolar membrane is called tonoplast. That is semi-permeable. Do transport event against concentration gradient. Important item. Into the vacuole, showing concentration in the vacuole is higher than cytoplasm. Vacuolar formation. Then different types of vacuole: contractile vacuole, foot vacuole, gas vacuole, otherwise called pseudo vacuoles. After that, 5 theory question, 5 NEET model question as usual. And if you like this episode, subscribe or like it, share it, comment on it. We can meet later. Take care. Bye. Meanwhile, short question I'll be putting after 9:30 every day at least 2. So we can meet later. Take care. Bye.